Why do patients with nephrotic syndrome develop low plasma protein levels? By the end of this video, you'll know the answer to that question. But first, let's start with the basics. What exactly are plasma proteins? Plasma proteins are proteins found in the liquid portion of the blood, the plasma, which is made up of about 90% water. There are three main groups of plasma proteins: albumin, globulins, and fibrinogen. Now that we know what they are, the next important question is, where are these proteins produced? Plasma proteins are mainly produced in two major sites. The first, and most important, is the liver, which synthesizes the majority of plasma proteins, including albumin and fibrinogen. The second source is the immune system, more specifically, B lymphocytes producing immunoglobulins. But production is only one side of the story. Plasma proteins are also catabolized in the body. This happens in three main sites. First, the reticuloendothelial system, mainly in the liver and spleen, where proteins are degraded. Second, the kidneys, where some proteins can be lost in the urine. And third, the intestines, where proteins can be eliminated through the feces. An important concept to remember is the relationship between protein synthesis and protein loss. If you understand this now, many topics later in medical school will become much easier. When a disease affects the organ responsible for protein synthesis, the production of plasma proteins decreases. As a result, the levels of plasma proteins in the blood also decreases. For example, in liver disease, the liver cannot synthesize proteins efficiently, which leads to reduced plasma protein levels. On the other hand, diseases that affect organs responsible for fluid balance, such as the kidneys, can also change plasma protein levels. For instance, if a person loses large amounts of water through urine, the body can become dehydrated. When dehydration occurs, there is less water in the blood plasma. This causes the proteins in the plasma to become more concentrated, leading to higher plasma protein levels. So in this case, the proteins are not increased because the body produced more of them, but because there is less water to dilute them. Plasma proteins are classified using electrophoresis. Electrophoresis is a method of identifying and separating plasma proteins by size using an electrical current. The results of an electrophoresis are shown in a graph, and they are albumin, alpha-1 globulins, alpha-2 globulins, beta globulins, gamma globulins. Fibrinogen is also a plasma protein, but is not measured using electrophoresis. Let's start with albumin, the most abundant plasma protein in the blood. Albumin is synthesized in the liver. Structurally, albumin is a simple protein, meaning it consists of a single polypeptide chain with a relatively low molecular weight compared to many other plasma proteins. At physiological pH around 7.4, albumin carries a negative charge, which plays an important role in many of its biological functions. Albumin has several key roles in the body. First, it maintains oncotic or osmotic pressure in the blood, helping keep fluid inside the blood vessels. Second, albumin acts as a transport protein, carrying many substances through the bloodstream. These include bile acids, calcium, bilirubin, hormones, and fat-soluble vitamins such as vitamin A, D, E, and K. Third, albumin serves as an amino acid reserve, meaning it can act as a storage form of amino acids. Fourth, it contributes to the buffer system of the blood, helping maintain a stable pH. And finally, albumin also has antioxidant properties, helping protect tissues from oxidative damage. All of this information must be overwhelming. What can we do about that? I have a great idea. Imagine the bloodstream as a vast ocean, and albumin as one of its most important components. At the surface of the water, albumin helps maintain oncotic pressure, keeping fluid inside the blood vessels. Moving deeper, we see a hook, representing albumin's role in transporting molecules, such as hormones, fatty acids, and drugs throughout the bloodstream. At the bottom of the ocean, there is a treasure, symbolizing albumin as a reservoir of amino acids that the body can use when needed. Covering the treasure is a net, representing albumin's role as a buffer system, helping stabilize the pH of the blood. And when the sharks approach, representing harmful oxidative molecules, albumin also acts as an antioxidant, protecting the body from damage. Now let's look at conditions where albumin levels change. An increase in albumin, known as hyperalbuminemia, is most commonly caused by dehydration, when water intake decreases and plasma becomes more concentrated. On the other hand, hypoalbuminemia or decreased albumin levels can occur in several situations. For example, it can occur during pregnancy or during hyperhydration, where increased water intake dilutes plasma proteins. Another cause is increased protein loss, such as in nephrotic syndrome, where albumin is lost through the kidneys or in conditions like severe wounds, burns, or protein losing enteropathy of the intestines. Hypoalbuminemia can also result from increased catabolism, which occurs during trauma, severe infections, or inflammation. Finally, decreased albumin levels may occur due to reduced synthesis, particularly in liver disease or in cases of malnutrition and malabsorption. Now let's move on to globulins, specifically the alpha 1 globulins. One important protein in this group is the alpha 1 antitrypsin, often abbreviated as AAT. Alpha 1 antitrypsin is synthesized in the liver and is considered an acute phase protein, meaning its levels increase during inflammation. Its main function is to inhibit different serine proteases, particularly elastase. Clinically, increased levels of alpha 1 antitrypsin are usually associated with inflammatory processes. On the other hand, decreased levels can lead to lung and liver diseases, which can affect both children and adults. Another important alpha 1 globulin is alpha fetoprotein, abbreviated as AFP. AFP is first produced by the yolk sac and later by the fetal liver during development. It is able to cross the placenta, meaning it can enter the maternal circulation during pregnancy, which is why it is commonly measured in maternal blood tests. In addition, AFP has been shown to induce apoptosis in tumor cells, partly through the activation of caspase 3. Now, let's look at the clinical significance of alpha fetoprotein. First, during pregnancy, AFP plays an important role in prenatal screening. AFP produced by the fetus can enter the maternal circulation, so it can be measured as maternal serum AFP. This test is commonly used to screen for fetal abnormalities. If AFP levels are increased, this may suggest the presence of congenital malformations. On the other hand, decreased AFP levels can be associated with chromosomal abnormalities. Now, outside of pregnancy, AFP also has important clinical uses. In non-pregnant individuals, AFP can act as a tumor marker. It is particularly associated with liver cancer as well as cancer of the gonads, including the ovaries and the testes. Now, let's move on to the next group, the alpha 2 globulins. One important protein in this group is ceruloplasmin, often abbreviated as CP. Ceruloplasmin is synthesized in the liver and is also considered an acute phase protein. Its main role is the transport of copper in the blood. In addition, ceruloplasmin acts as a peroxidase, which means it helps convert iron into a form that can be transported in the bloodstream. Because of this, it also plays a role in iron transport and metabolism. Ceruloplasmin also functions as an antioxidant, helping protect tissues from oxidative damage. Clinically, increased levels of ceruloplasmin can be seen in conditions such as pregnancy, lymphoma, which is a type of blood cancer, and inflammation. On the other hand, decreased levels can occur in copper deficiency, liver disease, and Wilson's disease, a condition characterized by excess copper accumulation in the body. Another important alpha 2 globulin is haptoglobin, abbreviated as HP. Haptoglobin is also an acute phase protein, and its main function is to bind and transport hemoglobin released from red blood cells. During inflammation, haptoglobin levels increases. However, in hemolytic anemia, where there is destruction of red blood cells, haptoglobin levels decrease because it binds the released hemoglobin and is subsequently removed from circulation. Next, we move on to the beta globulins. An important protein in this group is transferrin, also known as siderophilin. Transferrin is synthesized in the liver and is classified as a negative acute phase protein, meaning its levels tend to decrease during inflammation. Structurally, transferrin is a glycoprotein. Transferrin carries iron that is absorbed from the intestine and transported to several tissues, including the liver, spleen, muscles, and the red bone marrow, where it is used for hemoglobin synthesis and red blood cell production. Clinically, transferrin levels can increase in situations such as iron deficiency, pregnancy, and during estrogen-based therapy. On the other hand, decreased transferrin levels can be seen in thalassemia, which is a genetic disorder affecting hemoglobin, as well as in liver disease and in inflammatory conditions. Next, we'll talk about C-reactive protein, commonly abbreviated as CRP. CRP is synthesized in the liver and is considered one of the most important acute phase proteins. Its main role is to help the immune system recognize and eliminate non-self structures, meaning substances that do not belong to the body, such as bacteria or other pathogens. It does this mainly by activating the complement system, which enhances the immune response and helps destroy these foreign invaders. Because CRP is an acute phase protein, its levels increase significantly during inflammation. Clinically, elevated CRP levels can be seen in many conditions, including acute and chronic inflammations, bacterial infections, severe trauma, and neoplasm, meaning solid tumors. CRP levels may also increase during acute myocardial inflammation, such as after damage to the heart tissue. In addition, elevated CRP can also be observed in autoimmune diseases, for example, Crohn's disease. For this reason, CRP is widely used in medicine as a marker to detect and monitor inflammatory processes in the body. Moving on, we'll talk about fibrinogen. Fibrinogen is synthesized in the liver and plays a major role in blood coagulation. Structurally, fibrinogen is a hexamer, composed of 2 alpha chains, 2 beta chains, and 2 gamma chains. Fibrinogen is also an acute phase protein. In the coagulation cascade, fibrinogen functions as coagulation factor 1, and in its circulating form, it is inactive. During clot formation, the enzyme thrombin, which is active factor 2, cleaves fibrinopeptides A and B from fibrinogen. This process converts fibrinogen into fibrin, which initially forms soluble fibrin strands. Then, factor 13 stabilizes the fibrin by cross-linking the strands, converting it into insoluble fibrin, which forms the stable blood clot. Finally, we move on to the gamma globulins. Gamma globulins are immunoglobulins, also known as antibodies, and they play a key role in the immune system. These proteins are synthesized by plasma cells, which originate from activated B lymphocytes. Their main function is to bind antigens, meaning foreign or non-self molecules, and help the body defend itself against pathogens such as viruses, bacteria, and parasites. Immunoglobulins are tetrameric molecules, meaning they are made up of 4 chains. To simplify the structure, we use H for heavy chains, L for light chains, V for variable regions, and C for constant regions. Each immunoglobulin consists of 2 heavy chains and 2 light chains. Each chain has a variable region and a constant region. The chains are linked together by disulfide bonds, also called sulfur bridges. These bonds allow a certain degree of flexibility, so the molecule can change shape and bind effectively to antigens. At the top of the molecule are the variable regions. This is the antigen binding site, and it is highly variable. This entire top portion is called the FAB fragment, or the antigen binding fragment. At the bottom of the molecule is the constant region of the heavy chains. This part interacts with immune cells such as macrophages. This region is called the FC fragment, or crystallizable fragment. Immunoglobulins are classified according to the type of heavy chain they contain. These include immunoglobulin A, which contains the alpha heavy chain, immunoglobulin D, which contains the delta heavy chain, immunoglobulin E, which contains the epsilon heavy chain, immunoglobulin G, which contains the gamma heavy chain, immunoglobulin M, which contains the mu heavy chain. Together, these immunoglobulins form an essential part of the body's adaptive immune defense. Immunoglobulins can also be classified according to their light chains. There are 2 types of light chains, lambda and kappa. Now let's take a closer look at immunoglobulin G, one of the most important gamma globulins. It is divided into 4 subclasses, which are immunoglobulin G1, 2, 3, and 4. The main role of immunoglobulin G is in the secondary immune response. This means it is produced when the body encounters a pathogen for the second time, allowing the immune system to respond faster and more efficiently. Another very important property of immunoglobulin G is that it can actively cross the placenta. Because of this, immunoglobulin G antibodies from the mother enter the fetus circulation, providing passive immunity to the fetus and the newborn during the first month of life. This transfer of antibodies helps protect the newborn before their own immune system is fully developed. Next, let's talk about immunoglobulin M, another important type of gamma globulin. Structurally, immunoglobulin M forms a pentamer, meaning it is made up of 5 antibody units linked together. Because of this pentamer structure, immunoglobulin M can bind up to 10 antigens at the same time, making it very effective at capturing pathogens. Functionally, immunoglobulin M plays a major role in the primary immune response. This means that it is the first antibody produced when the body encounters a new infection. For this reason, the presence of immunoglobulin M in the blood often indicates a recent or ongoing infection. Unlike immunoglobulin G, immunoglobulin M does not cross the placenta, so it does not provide passive immunity to the fetus. Let's now look at immunoglobulin A. Structurally, immunoglobulin A forms a dimer. It is mainly found in mucosal secretions such as nasal secretions, bronchial secretions, intestinal secretions, saliva, and maternal milk. Because of this distribution, immunoglobulin A plays an important role in protecting mucosal surfaces against pathogens. It helps provide local immunity at sites where microorganisms commonly enter the body. Another type of gamma globulin is immunoglobulin E. Immunoglobulin E antibodies are also called reagens. They play an important role in type 1 hypersensitivity reactions, which include anaphylactic reactions. Elevated immunoglobulin E levels are commonly seen in allergic diseases, parasitic infections, and in some autoimmune diseases. Thank you for watching. This video was brought to you by MedPulse, created by medical students. If you found this helpful, follow for more simplified medical contents. See you in the next video.